Okay, jumping right back in. Sorry about that. I had to make a quick cut. But jumping back into part 2, we're going to do DNA-dependent RNA polymerases. So it adds ribonucleotide units to the 3' end of the growing RNA chain. Now, RNA synthesis is initiated, or it's started at specific DNA sequences called promoters. RNA polymerase binds at the promoter sequence about 10 bases upstream from the starting point of transcription. So it's kind of like we have a strand of DNA here. Remember, this DNA has already been separated. It has been separated into 2, so that way we can access the one gene. And right in here, we have a promoter region. The promoter region is just where it has the correct sequence to allow for the RNA polymerase to come in and to bind. So the RNA polymerase comes in and binds here and starts to travel down, and about 10 base pairs away, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, is where our actual gene is coded and where transcription will actually start. So this promoter sequence contains a consensus sequence composed of 5 from the 5' end, TA, TAAT. These are also known as TATA boxes, or PribNow box. I've almost never heard it called a PribNow box. I've almost heard it exclusively called a TATA box. So a second consensus sequence composed of 5' TTGACA, to the 3', so let's start on the 5' end, is localized upstream from the TATA box about 35 nucleotides, so 35 bases away from the TATA box. That's from the start point of transcription. A third AT-rich recognition element is also present between 40 to 60 in the promoter region in some highly expressed genes. Changes in the consensus region may influence binding of RNA polymerase and initiation of transcription. Kind of the main ideas to take from here, knowing what the TATA box, TATA or TATA, I'm trying to remember exactly how people pronounce it, knowing what that is, and that it allows, it helps allowing for the kind of where the promoter is, how we get the RNA polymerase to really bind and start doing its thing. And then we have a couple of other different regions. I wouldn't worry too much about all of those. Recognize kind of what they are, but don't try and memorize each of these different numbers. Understand that these different boxes, these different sequences, influence the binding of the RNA polymerase and initiation of transcription. So you can have these different regions, and like the location of the regions away from the gene are going to help determine how active that specific gene is. Because certain genes that produce certain proteins, our body's going to need more of those proteins, more of certain proteins than it is of other proteins. So we're going to our body's going to make sure that we have the proper expression of our proteins. Okay, so the elongation of RNA transcripts requires the unwinding of DNA. So the termination of transcription is signaled by a specific DNA sequence, AAAA, forming a hairpin loop that leads to the transcription of this section with ribonucleotide sequences of UUUU residues. Formation of this UUUU hairpin loop structure causes release of RNA polymerase. So the DNA is going to be transcribing, that's the right word, transcribing all the way down until it hits a DNA sequence of a whole bunch of adenine all stacked together, or all right next to each other. And by forming 4 U residues in a row, that causes a reaction that actually essentially pops it off. So, and by popping it off, it terminates and it stops it from continuing to transcribe down the DNA chain. and it forms a hairpin loop. We talked about hairpin loops just a little bit ago. Just a little bit ago about within the same chapter. So also, termination signal is recognized by the termination protein called Rho factor. At this point, binding of Rho factor to the termination site of the DNA template causes RNA polymerase enzyme and the RNA transcript to be released from the DNA template. So we have this combination of the quadruple U hairpin loop as well as the termination protein called the Rho factor to help end the transcription process and get that RNA off of the DNA to be able to go off and actually be used. So jumping more into transcription. So RNA has a base sequence that is complementary to one of the DNA strands. So this is where you can get some questions kind of similar. He could give you a DNA strand, something like this, probably a little bit shorter, and ask you what the complementary DNA strand is. Or he could give you this strand and ask you what the complementary RNA strand is. And just keeping in mind that the RNA now has the U's, which is what we want to look for. So, and when the RNA gets transcribed on the DNA and gets popped off and it goes off, as soon as it comes off of that DNA, it is what is called mRNA or messenger RNA. The messenger RNA is then going to go off and it's going to be read again. It's going to be read by the tRNA. The tRNA has codons and anticodons. I think we'll get into that a little bit later. But then we actually start each of these different chunks. Every 3 is called a codon, and that correlates to an anticodon on the the tRNA. The tRNA, and that's going to correlate to a specific amino acid. And we'll get more into that a little bit later on. Just wanted to give you a taste right now. Okay, so going back into transcription, we have introns, which are the non-coding regions of the RNA, and exons, which are the coding regions of the RNA. Both introns and exons are transcribed from DNA to RNA, and then RNA catalyzes the splicing of introns. I don't know if it gives a good example here. Kind of does. We'll get down to it. Or I'll just say, so introns, you can kind of think of it like we're going to make this whole strand of RNA, but in reality, the chunks of the RNA that code for that specific protein that we want are, it's like this chunk, this chunk, this chunk, and this chunk. These all together are what actually code for the proper protein, and those are our introns. These other spaces in between, you can kind of think of just like additional fluff. They have their own role, you know, kind of protecting the DNA strand, so that way those mutations don't occur. But the exon, sorry, let me correct myself. The pink are the introns. Introns are the non-coding regions. Non-coding means that we don't end up using them in the end. The exons are the coding regions. Let me just actually do this. Flip these. The exons are our coding regions. The introns are our non-coding regions. So literally what's going to happen is that these chunks are going to be spliced or cut. And the parts that we're going to use are going to be taken, and then all of those will be put together to form our what we actually need. And then these other ones, the introns, will be taken out, and they'll be kind of discarded. They'll be broken back up. Okay, and it should give us a good diagram a little bit lower in this chapter. Okay, so we have primary RNA transcript. So once transcribed, so we've transcribed the DNA into RNA, and now we have mRNA. So that RNA in eukaryotes is extensively processed. Splicing removes the introns and joins the exons, and it forms a contiguous sequence. Primary transcript is modified for protection from nuclease attack by 7-methylguanosine cap on the 5' end, and a poly A tail is attached to the 3' end. So this now spliced together mRNA that we've created, we need to protect it. So on the 5' end, we're going to put essentially this cap, this little protector on the 5' end, and then we just throw a whole bunch of A's on the end in order to protect it from essentially from getting broken down. The nucleases can end up breaking, or is looking to find these nucleic acids that aren't serving a purpose and break them down so that way they can reuse all of the components. But we don't want that to happen, so the methylguanosine cap and the poly A tail help to protect and prevent that from happening. Another little note, DNA-dependent RNA polymerase is inhibited by actinomycin D. Actinomycin D intercalates between the DNA strands and does not allow the RNA polymerase enzyme to move along for transcription. So it's kind of like put up a wall. It's going to grab on and not let the RNA polymerase pass to continue to transcribe. Okay. Processing of heterogeneous nuclear RNA. So this produces mature mRNA, which enters the cytoplasm through nuclear pores. So remember, all of this is happening, the creation of RNA, and all the DNA is stored inside of the nucleus. The nucleus have these little tiny holes, these little pores in them to allow stuff to go in and out. So once we get a fully formed mRNA, the proteins themselves are actually going to be formed outside of the nucleus, so that the mRNA is going to be transported and carried through those pores into the cytoplasm, where they can then be turned into proteins. Okay, but we're going to look all through here. So we have our different boxes. We have our Tata box. We have the cap site where we're going to put that methylguanosine cap, the protein start signal, and then we're going to have some splice sites, the stop signal, and then the addition of that poly A tail. And as soon as it's formed, we're going to have some splicing and some pulling things out, and then having some things put together. The cap goes on, we have the poly A tail, and then finally we have a fully formed mRNA. Let's check this chapter. Okay. Talking about transcription. So the RNA primary transcripts are produced, followed by the removal of introns, and finally produce a functionally mature mRNA containing that 5' cap with 7-methylguanosine. The number of times that has been repeated, hopefully by this point you've seen that and understand, okay, that's probably going to be important. So that attachment, and a 3' polyadenylate chain that's on the end. Again, remember, what's the purpose of both of those? It's to prevent the nuclease activity, so it's to prevent the nucleic acid from being broken down prematurely. Okay, so RNA polymerase II contains the enzymatic activity to attach 5' cap, which results from condensation of guanine with the triphosphate at the 5' end, followed by methylation by S-adenosyl methionine. Another enzyme, polyadenylate polymerase, adds the poly A tails at the 3' end. The process of removing the introns is known as splicing, and splicing is accomplished by a special complex structure having catalytic activity like enzymes and is called spliceosome. So let's make sure that we remember specifically our enzymes. We love to ask questions about the specific enzymes. that do these different jobs. Okay, and these spliceosomes, which is involved in the final formation of the mRNA from the primary transcript. All of these processes occur in the nucleus, or in the nucleus. So, we can see here we have the DNA. Again, this is the DNA, that polymerase 2, the RNA polymerase 2. Build all through. We have our cap. We have putting on our poly A tail. We have the exons. We have the introns, and those introns will be cleaved and cut out by these spliceosomes, and we end up with a mature mRNA. I do know that, to be completely frank, I feel like this specific chapter is a little bit confusing. It's not quite organized exactly the way that I would probably like it to be organized. So let's actually run over to see if we can find a video. So there are a couple of videos. Professor Dave here is a pretty good one. I've watched his in the past as well. A little bit shorter. If you want a more in-depth kind of running through everything, either looking at this video of probably Ninja Nerd, he should have one on this. Let's say it's probably included in this video. Yeah, there's these two different videos. There's the cell biology of the DNA transcription, which is a pretty long video. Here, go all the way down to RNA polymerase, but you can he has it broken up into different sections, so you can go and click through exactly what it is that you want to watch through. Stuff like the row dependent termination, maybe the transcription start site, those kind of points. I go through and watch these. Really good stuff. He'll do a very, very detailed walkthrough of it. But in all reality, you probably just need a surface level explanation. So Professor Dave would probably be a good one to just run through. Or Khan Academy. Khan Academy's another one that I use a lot. So I would recommend this one. This one probably DNA replication, RNA transcription, translation. I'm sure this is a really, really good video, and it's only 15 minutes. So I'd recommend spending a little bit of time going through some of these, using these resources. All right. I'm going to go ahead and get the last one recorded for you guys. I hope you guys have a great day.